Now? Okay, excellent. Well, welcome everyone to class. This is lesson number 17 on year 3 of science. And this is actually the last lesson that we have on cytology. So this is a very important lesson, and then we are going to transition to botany, to the study of plants. Okay, so let's start by having Maria teach and share with us what she has learned in the academic concept number 1. Okay, so Maria, the floor is yours.
Okay, thanks. So in academic concept 1, we learn about Edward Jenner, who is the one who discovered how to help people be immune to smallpox. He was born in 749 in England, and he became a doctor, and when he was small, he loved studying nature, and as soon as he was old enough, he went out there and studied the nature and the woods. And during his time, smallpox was definitely one of the deadliest diseases in his time, and it killed about 1 to 3 people who caught it. And if you were lucky enough to survive it, you were badly disfigured. He was very interested in smallpox, and he heard an old wives' tale that said that milkmaids couldn't get smallpox. And so he looked into the story and found out that milkmaids got cowpox instead. It's a weak version of smallpox that happens in cows, and the milkmaids, when they got these cowpoxes, they became kind of immune to smallpoxes. So through his observations, he tested his theory by taking some cowpox blister from a hand of a milkmaid whose name was Sarah, and he injected some of the fluids into a boy named James, who was just a neighborhood boy, and he just kept doing this injection over several days. It's increasing the amount of fluid to put inside James, and Jenner, he injected it with the smallpox virus at the end of the experiment, and the little boy, he became ill, but after a few days he was better. He had a full recovery and no side effects. And in the video, it shows that smallpox, it targets the skin cells, the spleen, lymph nodes, and the bone marrow, and you can get really itchy skin and spots that raise in your skin cells. And it also talked about how Edward wasn't the first one to find something to help people with smallpox, because before him, a long time before, there was a monk in China who crushed smallpox blisters, something like that, and she would take it and blow it into a healthy person's nose, and it actually kind of worked. But when Edward Jenner found out his vaccination that worked much, much better, they actually outlawed, I guess, kind of, that you couldn't do that procedure anymore of blowing that dust into yourself. And also in the video,
so that procedure is called?
That procedure is called variolation. But the reason why they kind of they kind of outlawed it is because still 3% would die from smallpox, and it was useful up until Edward Jenner found out his way. And yeah, and it also said that the smallpox and cowpox, the reason it helped is because they're actually from the same family. but cowpox is just a weaker version of smallpox, and it helps like fight your body fight off smallpox, the virus. And it was like he tested more and more on more people, and then it eventually his way of vaccination was accepted over time.
I have a question.
Mm-hmm.
Are chickenpox the same family as smallpox and cowpox, or are they in their own family?
Do you know that, Maria?
I do not.
Okay, so it is interesting. So, here we have cowpox, smallpox, and then chickenpox. Okay, so it basically it tells you which is the organism that type of virus becomes deadly. So cowpox is kind of deadly for the cows, but not for humans. Smallpox is deadly for humans, and chickenpox is deadly for chickens. Okay? They are similar in the sense that they all attack the epithelial tissues, basically the skin. Okay? And they all basically, when you get rashes and these poxes, all right, with pus. But the chicken pox, when the body develops immunity to chicken pox, it does not assure you immunity to smallpox, sadly enough. Okay. So the chicken pox, you can get chicken pox and still get smallpox.
Okay.
All right. Mm-hmm. So chickenpox is just, not deadly to us. It makes us sick, okay, but the body reacts soon enough to develop the immunity, and then you get better after, a week and a half, you are fine, okay. The problem was that smallpox didn't have, the body doesn't have enough time to develop immunity, and if you survive smallpox, the skin becomes, really deteriorated. Okay, all the poxes are still there. So this is actually a healthy child after surviving smallpox. Mm-hmm. Have you ever seen or met someone that had smallpox? You probably haven't because, well, I'm sure you haven't because when was the last time that we have a smallpox case in the world? The last recorded time when we had a smallpox, and that was part of, I think, this video, the documentary on smallpox. Mm-hmm. So it was 1977. So in 1977, it was the last case of smallpox. I remember when I was in elementary school in Argentina, the librarian in our school was a smallpox survivor. Okay, her case was not as bad as this one, okay, but she had a lot of marks on her face, okay, from smallpox. Mm-hmm. So yeah. So Maria, how was that? What is the question? What is the difference between then variolation and vaccination? Mm-hmm.
Well, vaccination takes a weaker form of the disease, um, and and it like helps fight off the stronger disease from your body and provide immunity from the same disease.
Mhm. Yeah. So I remember reading in the book on John Adams, John Adams, the second president of the United States. And during the time when John Adams was in Europe, okay, there was a smallpox outbreak in Boston, and his wife, John Adams' wife, got not the vaccination, but she got the variolation of smallpox. Okay, so she actually went to the doctor there in Boston, and the doctor would basically grab, actually, let's see if we can find, kind of the illustration here in the video. They would get the smallpox virus from a sick person, okay, so they would get it actually from someone that was sick with the smallpox, and then make scratches in the skin of someone that was healthy. All right. And Abigail Adams went that procedure. She was so scared of smallpox that she was willing to run the 3% risk in order to get the variolation procedure done. Okay. So Emmeline has a question. Her question is, why are you scarred after having smallpox? Why doesn't your skin heal? Is the skin just too damaged? Do you
Yeah,
know?
this yeah, the skin is too damaged. It's after having smallpox that it your body doesn't have enough strength to heal the skin. It's not like something where new skin grows and the old one falls off. It's kind of different. It's just it kind of stays in scars.
Mm-hmm. Yeah. And one of the big problems of smallpox is that this same, basically outbreak in the tissues of the skin happen also in all the tissues internally. Okay. So it happens in the lungs, in the esophagus, in the intestine, in the stomach, and that is why people die. That is actually why, very few survive, is because of all the scarring in all these tissues that, just the person cannot recover. Mhm. Okay. So Sydney says she looks like she got stung by a bunch of times by bees. Yes, but imagine this is permanent. Okay, and Liberty says that she also read that part too about the inoculation in Boston. Okay, yeah, very scary disease. So Edward Jenner was really an amazing person because he was able to develop this procedure to get the cowpox virus and inject it and then develop the immunity for smallpox. Okay. Now it is interesting. At this time, imagine he was born in 1749. Okay, so this happened, in the late 1700s. They didn't have any idea of why vaccination worked. They didn't know why it worked. They just tried it, and they knew that it worked, and they were like, okay, it works, let's do it. All right. Okay. Liberty asks, this is a child, will the scars stretch out and diminish some as they grow? I don't know. I really don't know. The librarian that I knew, that I had in my elementary school, she didn't have this many scars, okay, but she still had the scars, and they were not going away. That was her, So really, really interesting. Okay, so now let's go to the next scientist. Maria, thank you very, very much. You did excellent. Who would like to do the academic concept 2 on Louis Pasteur? I need actually someone also for number 3. Okay, all right. So anyone that would like to do number 2 on Louis Pasteur and then number 3, okay. All right, Maria could volunteer again. So Emmeline, if we need it, but I would prefer to spread the wealth around. Okay, Emmeline will do number 2. Okay, and so let me make sure, Emmeline, I'm going to unmute you. Okay. All right, there you go. Okay. If no one volunteers for number 3, Maria will have you gladly back. Okay. All right, Emmeline.
All right, so academic concept number 2 is about Louis Pasteur, and he basically proves the germ theory of diseases and perfects the process for killing bacteria, which is, pasteurization. So gives a little bit of a story. He was born in Dole, eastern France in 1822. He is described as very hardworking, but wasn't very considered, anything special. He was mediocre. He enjoyed art and singing, but then he found science, and he really enjoyed that, so he went to college and basically studied chemistry and became a professor. So it was while he was teaching that he met his wife, and he had 5 children. But during his time, the people believed
that about, I think that 2 or 3 of the 5 children died of typhoid fever.
Yeah, I know. I found that super sad.
Yeah, okay, but that was one of the things that motivated him to work on this, on all these issues of curing disease. Mm-hmm.
Yes. I thought I saw one quote one time where he said something like, how he was talking about the secret to how he found his goal and whatever, and he said that his strength lied in his tenacity and his, his passion and his interest in this subject. And I could see him losing his child as, being a great motivator for that. So, yeah, I found that admirable as well. So, going back, so people believe that bacteria basically appeared out of nowhere and that they just, up and came. They were spontaneous. And so he did several experiments to see if that was true. I know he tried something with grapes, and, that basically proved that the theory was wrong, that it wasn't spontaneous. But what I thought was funny was that, he had some skeptics. And so there was this academy, a French academy for sciences, and they were like, well, if you can prove that this theory is wrong, or if someone can prove that this theory is right, then they would give them 2,500 francs, which is like, what, 400 US dollars or something? And so he won the
a lot
prize for
more
that.
at that time. This was a sizable prize. Okay.
Yeah.
Yeah. Mm-hmm.
So I thought that was interesting and funny. But going back, while he was doing this experiment for the prize, he did this swan flask. So what would happen is he would apply heat, and then he would let the flask sit, and then there would be no bacteria. Versus when you apply the heat, remove the neck, and there's bacteria present, because we know that some bacteria need oxygen to grow. And then if you have the heat applied and you tilt it sideways, then there would be bacteria present, because it would just, I don't know, stay or whatever.
So this is a very interesting thing. Okay. So here we have the flask. Let me see. Go. I think that they have a picture here on the. on the article of the Flask itself. Okay.
Yeah.
All right, come on. I don't know why this is not coming. But the idea here is that this flask would have the neck sealed, okay? All right. And so when the heat was applied, what happened to all the bacteria?
Well, they get to a certain intense heat and then it just dies out.
Yes, okay. And so if you leave the flask with the neck seal, okay, and you just let it sit, the bacteria cannot get in, and so there's no bacteria growing.
Right.
So there's not really any spontaneous generation. There has to be some other way of the bacteria to get in. So here in the second one, he applied heat also again to kill the bacteria, but then remove the neck, and then the bacteria would basically just kind of get in and then grow in the liquid. Okay. And then the third part of the experiment, he did the same thing, but now he actually even let a little bit, just a little bit of the liquid get in contact with the air, and that would be enough to bring bacteria back in. Okay. So he basically was saying, you know, hey, bacteria is really almost everywhere, okay? But it's not that they are spontaneously generated. They don't just come out of nothing. And that was the big discovery. Okay. Griffin, you have a question? Let me unmute you. Okay, Griffin, what's your question?
I was just wondering, for our assignment, it says, right, wait, let me see. It says make a slide or create a slide.
For the assignment, let's talk about that after we finish. Is that okay?
Oh, okay.
Yeah. Okay. And then Liberty has also another question. She says, I once heard that something about the way milk is pasteurized can make some people intolerant to it. Why would that be? Okay. I really don't know. I know that some people have milk intolerance or lactose intolerance, and basically is that they cannot digest the lactate sugar, okay? But galactose is the sugar, but you know, I don't know why that happens. Okay. Okay. And Sydney says that his uncle has lactose intolerance. There's a lot of people that have that. So, Emmeline, what was the conclusion then of this experiment that Pasteur did?
The conclusion was that everyone acknowledged that it was right and that bacteria has to come from something. It isn't just spontaneous. So he got the prize. Whoopee! So yeah. A win-win.
What else was another thing that Louis Pasteur worked on?
So he worked on the cowpox, or not the cowpox. He worked on the anthrax.
Anthrax. Yeah.
So what would happen was that sheep and cattle, so cows, would get this bacterial disease, and it would basically affect the skin and lungs. And as we learned from dinner, that it can be transmitted to humans, which can cause skin ulcerations and pneumonia and whatnot. So he became curious with this, and so he decided he was going to give them a vaccine, so like a weakened form of anthrax, and give some while they're healthy, so that way they couldn't have anthrax. So, yeah. It's pretty cool.
Yeah. So now, the interesting thing was that Pasteur was able to now use the same techniques that Jenner developed of vaccination, but start applying and developing it to other diseases. Okay, so it's the same, and that is why vaccination now is just a generic term for any type of vaccine. Okay, for all of them, not just for the vaccine that comes from cowpox, as in the times of Jenner. Yeah. What did he do about rabies? Rabies. How do you say that?
Yeah. Rabies.
Rabies. Thank you.
Yeah. So what happened with that is that rabies is also another type of virus. And we know it's a very deadly disease, and it's horrible, and it is often given to us by the saliva. So normally like a bite by a dog that's infected with that virus. And so he wanted to see if that would work, like what he did with the anthrax. And so he made a weak form of rabies from rabbits that died from rabies, and he left it out for 5 to 10 days, and as it dried, he injected the vaccine to the animal that had been infected with rabies, and then it began to fight the infection. So it cured the animal and caused it to survive. And so later on, when it was still kind of being developed, a family came in with their young son, who was about 9 years old, who had been bitten by a rabid dog. And he was kind of hesitant because he wasn't a medical doctor, he was a chemist, and he had invented it, but it had never been used on a human before, so he was kind of like, but the parents were kind of like, oh, just do it, just do it. So he injected him with the vaccine, and the boy lived. So that was pretty awesome and pretty neat.
Yeah.
And then I think the last thing that it mentioned was how he contributed to the field of antiseptics. So during, like facilities, medical facilities, there was a problem going on where what would happen is doctors would treat their patients, and there would be like no form of cleanliness. So they wouldn't like wash their hands before they worked on a patient. They wouldn't clean their tools after. And so people were dying because of that, because of all the bacteria that was spreading from person to person, which caused them to lose their lives. So Pasteur kind of, recommended to, wash their hands before, wash their hands and wash their instruments before, doing procedures. And so there were a lot of skeptics from that, but eventually, they acknowledged that it helped, and yeah, he kind of was the inspiration for Joseph Lister for Listerine. So I thought that was pretty cool as well. So.
Yeah, and it's interesting that, Listerine, we just call it Listerine, okay? It's kind of a common term nowadays. But it comes really from this guy, Joseph Lister, okay, that work with Pasteur.
Yeah.
Yeah. Okay. Sydney says that her uncle was bit by a raccoon that had rabies. Again, how is it? Rabies?
Rabies.
Rabies. Okay. See, the English vowels get me all the time. Sorry about that, guys. Rabies. Thanks. So, what happened to him, Sydney? Do you know? Your mic is muted. I know that, my grandfather was bitten by a dog that had rabies, and he had to undergo the vaccination procedure, okay, that Louis Pasteur invented, okay. So it's, it would have been a very short life, okay. And my grandfather wouldn't have ever had my mother or me or anything, so you can see the effects that these vaccinations and these treatments have over different generations. Okay.
Yeah. crazy.
Mm-hmm. Yeah. One of the interesting things about the rabies vaccine is that you don't get the vaccine while you are healthy. You
Yeah.
get the vaccine just in case if if you get bitten by a dog or a carrier, a potential carrier of the disease, because the treatment is pretty traumatic. The treatment for the vaccination is not as simple as just doing one vaccine. You have to actually get a series of vaccines every day for several days, all right? And so not everyone gets vaccinated on that. Mm-hmm. Sydney, do you have a comment on that? Okay.
Oh, I was just gonna say, it was my friend's dad that got bit by a raccoon with rabies, and all I know is that he had to go to the hospital and get the vaccines and stuff. That it was long and really hard for him that he wasn't able to go to work for a while.
Yes, so see, there you have another example, another case where the vaccine was applied after getting bitten by the rabid animal, in this case a raccoon. Okay, so these animals in the wild are cute and everything, but they can be very dangerous because many times the animals are carriers, okay, of the virus, and they transmit the virus, but it doesn't necessarily kill them. It kills dogs, okay. I don't know if it also kills raccoons or not, but there are lots of this type of virus that they are carried by the animals, but then they become deadly to humans. Okay. Jens asked a couple of questions about the experiment here, all right? And so he asked the question, was the neck here open or closed? My understanding is that the tip of the neck was open, all right? And so in the first experiment, the bacteria would be killed by the heat, but then the bacteria couldn't travel through the neck, okay? There's not enough air movement or space for the bacteria to travel. The bacteria may get all the way up to here, maybe here, but it would not be able to negotiate the whole neck. In the second case, now the flask is really open, and so the bacteria can readily get in and start growing back in the liquid. And then in the third experiment, you have that now, because the liquid is moved through the neck into the area where the bacteria can get in and not be killed by the heat, then just even a little bit of bacteria is enough to contaminate the sample again and grow the bacteria again. Okay, so that is kind of how I interpret the experiment. Does that make sense, Jens? Okay, let's see. Let me unmute Jens. Okay, is that better explained?
Yeah. We're doing the training, and it's our business that's being benefited by the
Okay, that may be something else related to. Liberty says, some people say that Listerine is bad because it kills all of the bacteria in your mouth, including necessary bacteria for breaking down food. And is that true or not? Okay, so it is true that these antiseptics like Listerine kill all the bacteria, okay? The thing is that usually the good bacteria grows back faster, okay? And so, and the good bacteria lives not only in our mouth, but also all through our digestive system. And so it is still enough, there's still a lot of bacteria to reproduce. That is why, it's not a big deal. The bad bacteria, the problem with the bad bacteria is the bacteria that lives, in between the teeth, and then they build plaque and they make the caries, and they decay the tooth and so on. That bad bacteria, if it gets all the way into our digestive system, then it can make you sick, and that is food poisoning. All right, so that is why you use Listerine as a mouthwash, but you don't swallow it. Okay, don't swallow it. Does that make sense? Okay. All right. Let's see, Maria says, I believe that no one really is the first to invent an idea or vaccination. Maybe there were others who thought of the idea or studied it, but never published their ideas. I think that every single genius who invented something useful for the world had help from others, or wasn't truly the first, but that is just my opinion. Okay, so this is a very interesting and important thing to consider. Okay, so for example, Isaac Newton, who was the father of physics, he said that he built on the shoulders of giants. Okay, so basically he was saying, I didn't create all these things. I just, expanded on things that other great people had already studied and discovered and so on. Okay, and we can see the same thing happening here, where you have that Jenner, okay, was not that he invented the procedure from nothing. There were other people, in China and in Turkey and in other places, so that they were, already had experimented and done things related to vaccination or the variolation procedure. So he just adapted and changed. The same thing with Pasteur. He adapted and changed the procedure of Jenner in order to apply it to other diseases. Okay, so that is really how it works. The science is really a progressive thing. Now, I also believe that all these scientists, and they would be probably the first ones to admit that they had inspiration from a higher source. Okay, and so many times these scientists would be readily acknowledged that they were inspired by others, or by the spirit, we would call it. Okay. All right. Emmeline, would you like to explain more about the process of pasteurization? What is that? What is pasteurization?
So what happens with pasteurization is that you have to heats the liquids and lets them cool, so then the microbes would be killed. And what would happen in return for that is that the beverages would last longer and were safer to drink. And when it came to milk, because he was trying to figure out, how to work with milk, it didn't destroy the protein, which helped, with that. So it was safe to do with milk as well, which was kind of how we get, milk and vinegar and wines and cheese and juices.
Yes, so very, very important process, and it really has helped us to be a lot healthier as people, this process of pasteurization. Justin is asking, what happens when you get rabies? Do you know what happens when you get rabies?
I think I do. So, like, what happens, to my understanding, is that you have this virus, and what happens is that the virus goes to your muscle, and I think it's pronounced like subcutaneous tissue or whatever.
Yeah,
So that's
subcutaneous. Yeah,
kind of what
below
happens.
the skin. Mm-hmm.
Yeah, it happens to skin. So yeah, it's not fun. And
Yes.
so then it causes inflammation of the brain, and there's a lot of symptoms that aren't very pleasant. So uncontrolled movement, inability to move parts of the body. There's confusion, and normally death, like we talked about, as a result for dogs and things.
Mm-hmm.
So yeah.
So see what
So
it says here. Violent movements, uncontrolled excitement, fear of water, okay, and an inability to move parts of the body, confusion and loss of consciousness.
yeah.
And death nearly always results. So it's a very, very virulent disease. Really, it attacks the central nervous system. Okay,
Yeah.
so really a bad, bad disease. Okay. Um, Sometimes, the disease is also called hydrophobia, okay, because people become really fearful of water, all right, and they make foam from the mouth and everything, really, really nasty. Mhm. Okay. Um, what else? Do you want to say anything else about the germ theory? Mhm.
I guess, we talked a little bit, about the theory and how nothing is like never really truly discovered. We kind of talked about that. And it is true to an extent, kind of like what you said, that anything that is good is revealed from God to man. And the germ theory that wasn't spontaneous had already been like a theory, but the problem with that was there was no proof, there was no microscopes, there was nothing to, you know,
Mhm.
for it to be explained. So that's what made Pasteur so awesome, was that he, basically proved that it wasn't spontaneous and that the theories that had gone before him were correct. So
That's
in a
right.
way, yes and no, he did and did not discover it at the same time. But yeah.
Yeah. Mhm. Yeah, that's right. So again, we can really safely say that every day of our life, it's affected by the discoveries that Louis Pasteur made. Every time that you drink milk, every time that you wash your hands or brush your teeth or uh, drink any of the beverages that have been pasteurized, juices and so on, they are all things that we owe really to this scientist, an amazing discoverer, really. Hmm. Okay, excellent. Who would like to do academic concept number 3 with Alexander Fleming? Okay. If we don't have any takers, we'll ask Maria to do it again. Okay. Maria, would you like to help us out again?
Yes, I would.
Thank you. You get more extra points.
Yay.
Yeah.
So the last concept is about a man named Alexander Fleming, who was the one who discovered penicillin, which is the first antibiotic. He was born in Scotland in 1881, so this is a bit further, and we're going in now. And he was going on vacation, and behind he had left a jar containing Staphylococcus, which is a bacteria that is responsible for a lot of diseases.
Mhm.
And when he came back, he found that there was green-yellow mold, which is a fungi, had contaminated the jar. And just before he got rid of it, he noticed that the Staphylococcus, I don't know how you say that, bacteria,
Staphylococcus.
okay,
Yes.
thanks,
No.
covered all of the dish except for the area around the mold, which was completely clear of the bacteria. And so he kind of was like, whoa, and he figured out something that was, as we say, bacterial, something that he found could fight against bad bacteria. And Fleming decided to grow mold in a more pure culture and found that it produced a substance that killed a lot of disease-causing bacteria.
Mhm.
He identified the mold as Penicillium genus, and after some months of calling it mold juice, he finally released it as penicillin in March 7th, 1929. He continued doing his experiments and finding out more about it, and found that penicillin was actually really hard to grow, and it was even harder to isolate the antibiotic. And so Fleming started having the impression that that was the problem of producing it in quantity, because it appeared to be really slow, and penicillin wouldn't be important in treatment infection. So finally, he abandoned penicillin in 1940, and after that, not long, Howard Florey and Ernest Boris Chain and Norman Heatley, they started up his researching and mass-producing penicillin, and they had funds from the U.S. and British governments, which probably made it a lot easier for them to grow it.
Mhm.
And they made lots um, of it, and this is like after the bombing of Pearl Harbor. And by 1944, they had enough penicillin to treat all the wounded with Allied forces. And the antibiotics saved a lot of lives during the Second War, and they called it their first secret weapon. Fleming was pretty happy about the part that they developed the penicillin, and he described it as the Fleming myth. And he was thankful and grateful that his ideas were transformed and were made into a practical drug. Fleming was the first to discover the properties of this active substance, so he was given the privilege to name it, and he named it penicillin. He also grew and kept and distributed original mold for the 12 years and continued until his 1940, so that he could help any chemist who had enough skill to make penicillin. But they say at the end of it that without Fleming there would be no Chain, and without Chain no Florey, without Florey no Heatley, and without Heatley there wouldn't have ever been penicillin.
Mhm.
And it's still used in our day, and we use it in our modern antibiotics and test similar methods to grow it. And in the reason it helped in World War II is because the worst thing during wars was infection. They didn't have anything to help the soldiers with pain and all that. So infection was the worst part of it. Even really small wounds, the soldiers would die from it. And that's kind of what Fleming was really desirous of, to find something like this. He wanted to because of all the soldiers out there on the battlefield dying because of it. So it kind of pushed him forward to discover the penicillin. But when he wasn't successful, he kind of just dropped it. But thankfully, there were some others who got it up and produced it.
Yeah. You know, think about it. This is really amazing. During World War One, more soldiers died of infections than of wounds, of bullets. Okay. You know, so your chances of dying were much higher when you got into the hospital. Okay, because if you got into the hospital, it was because there was some sort of wound or problem. Okay, and the chances were that you were going to die because there was no way of fighting the bacteria. They didn't know what to do. Okay. And so this discovery, really changed the outcome of World War II. Didn't affect World War One, but it affected World War II. And Fleming he was a doctor during World War One. And so he had seen the problems of infectious diseases. Okay. He had seen that firsthand, really, all the time there. Okay. And so this discovery really of penicillin was literally, okay, and I don't have any problem admitting it was a miracle sent from God to help the Allies. All right, because look at the timing of things. Okay, again, review the timing of these developments, Maria, for us.
So the timing was during his time. It was like during World War II, wasn't it?
Yeah. So when did he saw the culture with the mold? Okay,
1928, September 28th.
that's right. So
19.
28. So this is 10 years, 9 years after the end of World War One. Okay. When did he publish his discovery?
Um, he released it on March 7th, 1929, so a year after.
A year after, okay, half a year after or a year after. All right. Then he kept working on it, but he couldn't really do anything. All right. And then he basically gave it up when?
1940.
1940. What year is that? Was World War II already going on?
Um,
World War II started on September 1st, 1939, when
so it was
Germany
just a
invaded
year
Poland.
after.
Yeah, so it was right at the beginning of the war, okay? And then as England became involved in the war, these two scientists, Chain and Florey, and then Healy, they started really, it was a race to see if they could mass produce penicillin. Okay. And the US was they started helping on the production of penicillin in the East Coast, okay? And by the time that the invasion of Normandy happened, 1944, they had enough penicillin there at hand, all right? So how many people died during D-Day, during the invasion of Normandy? Do you remember how many Allied soldiers died? About. Mm-hmm.
I don't remember, but
Uh-huh.
probably a lot.
Yes, it was in one day, 10,000 soldiers died. Okay. How many people do you think, how many of those soldiers, how many were wounded?
Probably even more than that.
Exactly, yes, a lot more. Okay. But because they had penicillin, very few of the wounded died. They actually recover and continue the fighting. Okay. And the Germans didn't have penicillin at all. Okay. So can you see the hand of God in blessing the Allies for this? I think that is one of the most amazing miracles, really, because, Fleming could have thrown this away. He could have, just not pay attention to it, but he had enough training and he had enough of the prompting of the Spirit to look at this. Look at what happened. There's no bacteria. Why? There's something that the mold is doing to kill the bacteria, you know. So, but the timing is just literally miraculous. You cannot, ascribe it to anything different than a total miracle. Anything else that you would like to say, Maria?
Well, I'd just like to say the kind of like what you said, it's kind of just amazing how what the timing and everything, and how God can use how God can just use a fungi mold and to have a scientist realize what was going on and what he could do with it. It's kind of amazing how he made the earth that way, how he made bacterias, and how he created all of this for our use, for us to invent one day.
Absolutely. Absolutely. Really, you can really see the hand of God in all of these things. To me, is so inspiring, frankly. Yeah. Nathan, would you like to take us through the gospel principle and how this gospel principle applies or relates to this lesson?
Yeah, sure. Well, the gospel principle is the work of Joseph Smith has increased our understanding of truth and has blessed our lives in countless ways. So, without what I think is without Joseph Smith, nothing could have happened because, well, I'm not sure if Joseph Smith didn't want to do the work, if God could find somebody else. But like these great scientists who contribute so much to the world, like the earthly world and how it works. Joseph Smith has been a church scientist. He has contributed so much to the church's ways. And so, D&C 135 is one of my favorite scriptures. And it's saying that, I'll read it to you. Joseph Smith, the prophet and seer of the Lord, has done more, save Jesus only, for the salvation of men in this world, than any other man that ever lived in it. In the short space of 20 years, he has brought forth the Book of Mormon. which he translated by the gift and power of God, and he has been the means of publishing it on 2 continents. He has sent the fullness of the everlasting gospel, which it contained, to the quarters of the earth. He brought forth the revelations and commandments which compose this book of doctrine and covenants, and made otherwise documents and instructions for the benefit of the children of men, gathered many thousands of the Latter-day Saints, founded a great city, and left a fame and name that cannot be slain. He lived great, and he died great in the eyes of God and his people, and like most of the Lord's anointed in ancient times, he has sealed his mission and his words with his own blood. So, with that, that scripture is saying that Joseph Smith has done more work than anyone in the church. He has saved no man other than Christ, has saved and prepared man for this time, because he went through all the trials. He went through jail twice. One of the times he didn't make it back alive. He got killed. He was driven from his home. He was spit on. He was treated in the worst ways, but he never lost hope because he knew that he was doing the right thing.
Mhm. And it is interesting to see that, these scientists, okay, they help literally save thousands and millions of human lives, okay, from disease. Joseph Smith is really helping us to get closer to Heavenly Father and save our spiritual lives.
Mm-hmm.
Yeah. When you consider about all these things, how do these lessons, Nathan, apply to you in your life? How do the examples here of these four men apply to each one of us?
Well, to me, it was like they didn't like succeed on their first try, and if they would have given up, we wouldn't have had the vaccine for this diseases. So it just shows that you should always try your hardest. Like Thomas Edison said, I didn't fail. I just found out 1,000 ways how not to make a light bulb. He never gave up until he found the way. And that's the same with us. If we give up the first time we make a mistake, then we won't make it anywhere. But we're gonna make a mistake, and if we just get up there, then that's where we're gonna end.
Mhm. That's right. Mhm. Have you had a chance of watching this video, this documentary on the smallpox virus?
I watched a little bit of it, but I didn't have enough time to watch all of it.
Okay, so I really encourage all of you to watch it all because it is interesting. It goes through the whole history of vaccination and everything, but then it tells you about how the disease got eradicated from the whole world. And these people here in the video are literally the people that made it happen. Okay, so some of these scientists that appear in the video are the ones that actually were there doing the work and the vaccination to make it happen. And so it's not just the people and thousands of other volunteers that did that. that eradicated this disease from the planet. That is an amazing story, really. And what that tells me is that, I don't have to be a great scientist, okay? Sometimes I just need to do my part, all right, in helping others do the right thing. So I am not going to be Joseph Smith, but I can be, a good missionary and share the gospel with those around me. Okay. So that is kind of one of the things that I learned, that I can do my part, my mission. Mhm. Emmeline put here a quote about Alexander Fleming and says, I've been trying to point out that in our lives, chance may have an astonishing influence. And if I may offer advice to the young laboratory worker, it would be this: never to neglect an extraordinary appearance or happening. So really to pay attention, okay, to pay attention, even the things that you are learning here in these lessons, you don't know how much is going to affect your life, okay? So, all the things that we learn and study and everything may have a huge impact later on. So we need to really pay attention to all these things, okay? Thank you very much, Nathan. Chas, would you like to dismiss us with a word of prayer?
Do you hear me?
Yes, go ahead.
Okay. Dear Father, for you to say, thank you for everything that you've done for us. Thank you for all us being able to learn this, and that we'll all use all this teachings in our mind, and that we'll study it more, and that we'll do good on our homework, and that we'll do good in this weekend. In Jesus Christ, amen.
Amen. Thank you very, very much.